During digestion, complex carbohydrates are hydrolyzed into monosaccharides, primarily glucose. The catabolism of glucose is the primary source of energy for cellular production of ATP. The anabolism of glucose is also important in order to regulate blood glucose levels. Let's learn about the glucose catabolism. The generation of ATP for use by cells involves 4 enzymatic pathways. These are glycolysis, formation of acetyl coenzyme A, the Krebs cycle, and the electron transport chain. Because glycolysis generates ATP in the absence of oxygen, it is considered an anaerobic pathway. To generate numerous ATP molecules, oxygen is needed. The remaining 3 pathways have varying concentration of oxygen and are considered to be aerobic pathways. Let's explore each one of these pathways. We begin with the glycolysis. Glycolysis is the first step of glucose catabolism, and it occurs in the cytosol of a cell. The first part of glycolysis involves phosphorylating glucose and then breaking it into smaller phosphate molecules. The second part of glycolysis is the oxidation of these smaller phosphate molecules, thus producing reduced coenzymes and some ATP. As a result of glycolysis of a single glucose molecule, 2 ATPs are produced and 2 reduced coenzymes. Much more energy is still in the 2 pyruvic acid molecules that remain from the single glucose molecule. Let's learn about the fate of these pyruvic acid molecules. Many more molecules of ATP can be generated from the energy still stored in the pyruvic acid molecules. However, this energy can only be harvested if the cell is well oxygenated. If there is inadequate oxygen, the pyruvic acid will be converted into lactic acid. However, if there is adequate oxygen, the pyruvic acid will be further harvested for much more energy in the mitochondrion of a cell. Cells that lack mitochondria generate significantly fewer ATP molecules. Let's continue to learn more about the glucose catabolism, now in the mitochondria. During the second step of glucose catabolism, which is the formation of acetyl coenzyme A, each of the 2 pyruvic acid molecules resulting from the glycolysis is oxidized to produce a reduced coenzyme. In addition, each pyruvic acid molecule loses a carbon dioxide molecule to form acetyl group. Each acetyl group will bind with a coenzyme A to form acetyl coenzyme A. Acetyl coenzyme A now will be taken further into the Krebs cycle for continuation of the glucose catabolism. During the Krebs cycle, the glucose molecule is completely oxidized. 4 more carbon dioxide molecules are released for a total of 6. 2 energy molecules are produced during the Krebs cycle. The remaining energy is in the acetyl groups is transferred to reduced coenzymes. The primary function of the Krebs cycle, therefore, is the production of many reduced coenzymes. It is in these molecules that most of the energy harvested from the original glucose is now resting. Let's see what happens to the NADH molecules in the next step, which is the electron transport chain. During the last step of aerobic glucose catabolism, the reduced coenzymes are used to generate ATP. This step occurs in the inner membrane of the mitochondrion. When the reduced coenzymes are oxidized, the energy is used to pump hydrogen ions across the inner mitochondrial membrane. In this process, a byproduct is formed, which is water, and that is when the hydrogens combine with oxygen. Hydrogen ions are slowly accumulated and form a concentration gradient between the inner membrane and the matrix of the mitochondrion. The concentration of hydrogen ions provide the force to produce ATP molecules. As the hydrogen ions diffuse down the concentration gradient across the membrane, they pass through the protein that synthesizes ATP. The process of producing ATP using the movement of hydrogen ions down the concentration gradient through ATP synthase molecule is known as chemiosmosis. Let's summarize the ATP yield from the glucose catabolism. As we've seen so far, the goal of glucose catabolism is to harvest energy from fuel molecules to produce ATP. In turn, the energy in ATP is used by the cell to perform its functions. And also we've seen that other products of glucose catabolism include production of water and 6-carbon dioxide molecules. We can see here in the summary that the cell can produce up to 38 ATP molecules from a single molecule of glucose. Now let's learn a little bit about the glucose anabolism. That means making of the glucose molecules. Glucose can be formed through two different anabolic reactions, glycogenolysis and gluconeogenesis. These reactions help to regulate blood glucose. So let's explore each one of these, beginning with the glycogenolysis. Glycogenolysis is a process that breaks up glycogen and occurs in the liver and in skeletal muscles. The hormones epinephrine and glucagon stimulate glycogenolysis. As the glycogen is hydrolyzed, blood glucose will increase and may be oxidized by the cells to get energy. When glucose is needed, glycogenesis is stimulated by the hormone insulin. As the glycogen is synthesized, blood glucose will decrease, will be removed from blood and stored in the liver and in the skeletal muscle cells. Finally, let's explore how glucose can be formed from non-carbohydrate sources. Non-carbohydrate molecules such as lactic acid, amino acids, and glycerol can be converted into glucose in the process of gluconeogenesis. Like glycogenolysis, gluconeogenesis also occurs in the liver. The hormones glucagon and cortisol stimulate gluconeogenesis, and as the glucose is synthesized, blood glucose will increase.